This study presents a protocol for the differentiation of neural precursor cells solely induced by direct current pulse stimulation in a microfluidic system. This technique provides a beneficial approach for reducing the experimental setup time, sample volume, and reagent volume. Furthermore, the MOE chip is suitable for confocal microscopy observations. The simple direct current pulse treatment can control mouse neural precursor cells' fate and could be used to develop therapeutic strategies for nervous system disorders. To begin, draw patterns for individual PMMA layers and the double-sided tape using appropriate software. Switch on the carbon dioxide laser scriber and connect it to a personal computer. Open the designed pattern file using the software. Place the PMMA sheets or double-sided tape on the platform of the laser scriber. Then focus the laser onto the surface of the PMMA sheets or the double-sided tape using the autofocus tool. Select the laser scriber as the printer, then use the laser scriber to start the direct ablation on the PMMA sheet or double-sided tape. Remove the protective film from the PMMA sheets and clean the surface using nitrogen gas. For bonding together multiple layers of PMMA sheets, stack 3 pieces of 1 mm PMMA sheets and bond them under a pressure of 5 kg per square centimeter in a thermal bonder for 30 minutes at 110 degrees Celsius to form the flow or electric